Assalamualaikum and a very good day to my honorable panels. My name is Eza Nurizni Ain binti Izal Ain. I will present to you my Bachelor Degree Project 2 with the title Evaluation on Antioxidant Capacity of Malaysia Herbal Plant as Potential Wound Healing Therapy. My supervisor is Technology Dr. Siti Aisyah Manso and my co-supervisor is Technologist Dr. Adif Hisham Iraza. The purpose of my study is to identify antioxidant compound in medicinal plant as wound healing properties. Also, this study will evaluate the acceptance of consumer towards herbal cream. Three objectives were to conduct for this experiment. First is to identify bioactive compound related to antioxidant substances in medicinal plant for wound healing. Second is to analyze the potential mechanistic pathway of natural antioxidant substances in wound healing. And lastly, to evaluate the acceptance of consumer towards herbal cream. The reason on why the study is important is because nowadays emerging diseases have caused tremendous change in medical treatment. For example, wound care and its treatment. The infection is a major problem provided with impaired wound healing conditions, had prolonged in tissue regenerations. These conditions may be varied according to specific complications faced by the patients, such as diabetes mellitus, eczema, and hemophilia. Wound healing consists of four phases, starting with hemostasis, inflammations, proliferations, and lastly, remodeling. However, during inflammation phase, the overproduction of rows of free radicals might cause oxidative damage, therefore lead to detrimental effects towards tissue regenerations. They also pose the risk of prolongations in inflammation phase, which cause severe pain, redness, and swelling in injury area. To understand the nature of free radicals, these reactive molecules tend to steal electrons from other molecules which has caused changes in an important molecule change such as protein. Thus, the role of antioxidants came into this process as agents to reduce and suppress the excess free radicals. Antioxidants will act as a reducing agent by donating electrons to quench the free radicals which missing its electron. In this study, we selected 52 eligible research papers for literature review among 116 hits of articles in primary research. These selected articles had been searched using several electronic databases. The use of hedonic tests and the collected raw data was received from third party, which was requested to be analyzed. Therefore, it must be noted that this study does not cover for fieldwork and constructions of questionnaire. For this survey, the scale and the total respondents are shown in Table 1 and Table 2 in the poster. In this study, gender factor is disregarded due to some of the respondents does not share their name. This study had chosen 6 selected Malaysian medicinal plants which are Chromolena odorata, pokok kapur terbang, Cinnamon cassia, which is kayu manis, Camphor galanga, which is seko, Curcuma longa, kunyit, Hibiscus rosa sinensis, which is bunga raya, and Hibiscus subdarifa, which is rosa. This plant exhibit antioxidant capacity due to the presence of flavonoids, polyphenols, commerce, and other compounds such as cinnamaldehyde. The identified antioxidant substances for this study was guanylate, quercetins, camphorol, curcumin, cinnamaldehyde, and chlorogenic acids. The potential of its mechanistic pathway in wound healing had been identified in nuclear factor adroit 2 related factor 2 or NRF2, which is an important transcription factor for expression of antioxidant response element or ARE and protein kinases. Four antioxidants were identified to induce NRF2 as natural activators which regulate oxidative damage by expressions of ARE or enzymatic antioxidants such as superoxide dismutase, peroxidosins, glutathione peroxidase, and catalase. It was also reported that NRF2 activations contribute to stimulations of cell proliferations and migrations during tissue formations. This mechanism is shown as following in Figure 2 in the poster. Lastly, the evaluations on consumer acceptance toward herbal cream consist of 5 formulated creams, which are VCO creams, cinnamon oil cream, C. odorata cream, lemon oil creams, and Cananga odorata oil creams. The data analysis had concluded that the hypothesis of this study is overall acceptability of herbal cream depended on age factor, and it is found that the herbal cream received a high trend of favorability for overall herbal creams sampling is from group of age at 20 to 29 years old. As conclusion, all the objectives were achieved. A few recommendations were suggested, such as the study of scatalene and chlorogenic acid compounds as NRF2 activators in vitro cell-based assay. Also, the evaluations on consumer acceptance toward physical characteristics of herbal cream with solid questionnaire information should be conducted again. That's all for my presentations of my bachelor degree projects too, with the title Evaluations on Antioxidant Capacity in Malaysian Herbal Plant as Potential Wound Healing Therapy. Thank you and a good day.